Most of the applications so far have been to model organisms, and in some cases it's really made things possible that weren't before in model organisms. We started out working on Drosophila. At the time, there wasn't a gene targeting technology in Drosophila, but the person who taught me about Drosophila, Kent Golick, and his student Yikong Rong, published a gene targeting procedure for Drosophila before we got ours done. But we were able to add a lot of efficiency to the approach that they had developed. So I think the model organisms where this is being used most now are zebrafish, where there was no way to make targeted mutations prior to this. There wasn't even a very robust transposon technology for zebrafish. So the zebrafish people have jumped right on this. It's very easy to do the ZFN mutagenesis in zebrafish. You just make messenger RNAs for the pair of proteins for your target, inject those into embryos. The embryos are big. People do message injections into embryos every day in a zebrafish lab. And so this is just doing something they're very comfortable with, and then screen down the road for the mutations that you're looking for. The other organism where it seems like this is taking off is the rat. And the rat has been playing second fiddle to the mouse in terms of experimental genetics for the last 20 years or so, partly because of the wonderful mutant collection that the Jackson Lab holds, but then more recently because of the embryonic stem cell technology and the ability to make targeted knock-ins and knockouts using the procedures that Mario Capecchi developed for gene targeting in ES cells. So the mouse was dominant, right? No way to do targeted mutations in the rat, no ES cell technology, no gene targeting. And now that people have shown that you can do gene targeting, at least targeted mutagenesis, by, again, injecting messenger RNAs into rat embryos, that's excited the rat community, as I understand it, third or fourth hand, because now they can make mutations in genes of interest, and they can use some of the size and physiological advantages of the rat to study things that weren't so easy to study in the mouse. So those are the two experimental organisms that I think have gotten the greatest benefit from the ZFN technology. The other thing that's happened is that people have started to think about gene targeting as a real possibility. You know, what's my organism? What's my gene that I'm particularly interested in? Do I have to wait around until a natural mutation occurs? No. Now I can think about making a targeted mutation in my gene of interest, maybe even making a targeted gene replacement where I put in a sequence that I want to see in that gene rather than the one that nature has given me. And so just the fact that the technology is out there and people are beginning to realize what the capabilities are, I think has changed the way people think about model organisms. We have a collaboration with someone that's just recently demonstrated that the ZFNs work well in silkworm. So there's a model, but also a commercial organism. I know there are people working on mosquito, looking toward abatement of these disease-carrying vectors. Essentially any organism, if you can figure out how to deliver the ZFNs and be able to screen for whatever changes you want, the technology doesn't rely on anything that's specific to any particular organism. Double-strand breaks are repaired with the same by the same pathways with the same machinery in essentially all organisms. So you make a break, you're going to get mutations. If you can figure out how to do it right, you can get the gene replacements by homologous recombination. So I think the people who are aware of the technology think about how to do genetics in a different way. In the future, the applications beyond expanding into other genes in current organisms and into other model organisms, the prospects for human gene therapy seem to me to be pretty wide open. Sangamo, in collaboration with people at University of Pennsylvania, have a clinical trial going on now using ZFNs to knock out the HIV co-receptor CCR5, and they, as far as I'm aware, they've only been able to talk about one patient who's received his own hematopoietic stem cells back after treatment to knock out CCR5. But it looks like things are working at the level they would have hoped, and I hope that future patients in this phase 1 trial will show the same sort of thing. But any situation where you can do an ex vivo treatment and then put cells back into a person seems to me is ripe for ZFN-mediated gene manipulations. So right now, hematopoietic stem cells, or T-cell precursors, I guess they're actually doing T-cell precursors, not the most primitive stem cells. Those things are obvious candidates because it's well known how to get these cells from bone marrow and culture them productively and reimplant them in the patient. Other types of stem cells are still being evaluated, and I think it's too early to say whether we'll eventually be able to treat traumatic nervous system injuries with neuronal stem cells of some type, or advanced neurodegenerative disease with neuronal stem cells of some type. But those are things that you could think about. And the ZFN tech, anytime there's a genetic change that might be advantageous, you can think about using the ZFN technology to do that. The other thing I know people are excited about is modification of crop plants. So Dow AgroSciences has a big program. They've made a large financial commitment to this technology in collaboration with Sangamo. And there's obviously genetically modified crop plants out there now. There's some resistance to that. I know that scientists at Dow Agro are hoping that targeted modifications, rather than putting in transgenes, but very targeted modifications of existing genes, might be more acceptable to the public and more attractive to the growers. So that's something where and I'm not a plant biologist, but you can imagine making subtle changes that would confer resistance to herbicides, resistance to drought, resistance to higher electrolyte concentrations in soil, that sort of thing. So I went to a meeting a few years ago where people were talking about different methods for genetically modifying mosquitoes in the laboratory, introducing them into wild populations, and having genetic modifications in those lab mosquitoes transmitted through whole populations, and have those modifications be ones that would either make the mosquitoes sterile or make them resistant to the life cycle of the malaria organism or some other parasite that these mosquitoes transmit. And since there are quite a number of these mosquito-borne diseases, if people can figure out ways to make these genetic modifications that would allow the mosquitoes, the modified mosquitoes, to remain genetically robust so that they can transmit these modifications into populations, then you have the possibility of modifying wild populations by this ingression of modified genes. So